We have 700 plus registrations.
Okay.
Yeah.
Is this the highest number you have so far, or you had before that also?
So far, this one's the highest, and let's see how many join
Yes, that
during the live.
50% attendance is generally what we expect.
Yeah, so we
Exactly.
are live now. We have 700 plus registrations.
Okay. So maybe you will have to mute YouTube live because I will keep hearing that as an echo. Whosoever has the mic on. Can you hear me?
Yeah, ma'am. So we are not going to have our mics on Zoom meeting. So we would also be here on YouTube directly. So there would not be any problem of echo.
Okay.
Yeah.
Sure, sure, sure.
Yeah. Correct.
Shantanu, can you just email, like WhatsApp me the link for YouTube live? The link which has been shared here?
Yes, sir.
So maybe we can start.
I guess we'll wait for 2-3 minutes and then we'll start.
Sure. Okay.
मेरे में सांस पहले ली तेरे में बाद में ली Let's start.
Yeah.
Hello everyone and good morning. Welcome to the talk on Introduction to qPCR for Disease Diagnosis by Dr. Shilpi Sharma. I'll introduce to our uh, professor. Um, she is Dr. Shilpi Sharma. She is an UGC assistant professor at Department of Biotechnology, Savitribai Phule University, Pune. She has completed her PhD from Institute of Genomics and Integrative Biology, Delhi, and she has also completed her postdoc research at University of Michigan and Chicago. We are glad, ma'am, that you could join us for this webinar today. I hope you enjoy it, and so does our audience. Also, I would like to point out that we'll be sending you links of our YouTube channel, Telegram group, and Instagram, and also our website. So feel free to subscribe to all our channels for further updates. And you can also join our Telegram group to get 50% off on upcoming webinars. Thank you. I'll handle the control to Dr. Shilpi Sharma.
Thank you, Shantanu. Thank you so much, LifeGenBio, for having me here. And I am really excited to be presenting a talk on the use of quantitative real-time PCR for disease diagnosis. So before I begin this talk, I would just like to make a small mention here. Actually, it's a dedication to Dr. Kary Mullis. He is the guy who invented the technique of PCR. And in fact, he got the Nobel Prize in Chemistry for this invention. And to be very frank with you, recombinant DNA technology would have not been possible if Kary Mullis did not do this very noble invention. And he truly deserved this Nobel Prize because talking about any field of science that you talk about now, it needs the application of real-time PCRs or PCRs, and we'll see how this technique has evolved over years. Into four different parts. The first would be understanding how a conventional PCR is different from a real-time PCR. What are the chemicals and instruments that have been used or that are in practice for doing real-time PCR? What are different chemistries which are used? How you can do these PCRs? How you analyze the data which is obtained from real-time PCR? And examples of uses of real-time PCR, we'll talk about disease diagnosis, we'll talk about SNP detection, we'll talk about methylation of DNA, and so on and so forth. So if you look at this image here, just to start with, this is actually how your real-time PCR results look like. This is actually a electropherogram of your real-time PCR, which is depicted here in the panel below. So we'll see how we come to these results, how we analyze these results over the next 1 hour. So if I talk about a conventional PCR, so initially when people wanted to amplify a part of the DNA, it was difficult. So people had to work with the entire DNA, and you had to cut down pieces of DNA which were longer than what was required using restriction and enzymes. And then people had to, it was a very tedious process. Identify a target sequence and then amplify it. So if you just look at this cartoon here, there are different genes which are listed here in this operon in a bacterial system. And I just want to amplify the TRPA gene. So what I can do is I will design primers which are spanning this TRPA gene. When I say that, I mean that which are at the boundaries of the TRPA gene. And using a polymerase chain reaction, I can make multiple copies in the order of several millions. Like the number of PCR cycles, the number of times you do a PCR would determine how many TRPA copies I would have in a tube, a very small tube, a 10 microliters or 20 microliters volume, right? So how do you do a PCR? We'll just go through that. This is a conventional PCR, and basically it deals with the selective amplification of a chosen region or region of a DNA. So say for example, this is your DNA region, and the region which has been highlighted here in blue is the TRPA gene, which we just talked about. Now, a conventional PCR would actually make multiple copies of this target sequence. We call this sequence that we want to amplify as a target sequence. And what do you really need here are your primers. Primers are nothing else but known sequence oligonucleotides, which you can get synthesized. And they are going to mask, like they're going to be at the boundaries of the target sequence to be amplified. Apart from that, you will require DNTPs, which are the building blocks of the polymerase chain reaction. And you require a thermostable enzyme. Because why a thermostable enzyme? Because there is a denaturation step and an annealing step, which is carried out at high temperatures. So you want an enzyme which will be stable at high temperatures. But now with the major technologies which have developed, you can also have enzymes which can work at room temperature or which can work at high temperature. So let's talk about the traditional DNA polymerases, the Thermus aquaticus derived Taq DNA polymerases, which were used and which are still in use for doing these reactions. So basically, this is a double-stranded DNA molecule. You first have to denature it. When I say denaturing it, I mean that you have to break the hydrogen bonds between the two DNA double strands, get them into single-stranded conformations. Then this is your denaturation step. After this, allow the primers to go and anneal to your templates. So when I say anneal, I mean that these short oligonucleotides, which are around 18 to 25 like base pairs in length, they will go and hybridize with the sequences where they're supposed to. After that, you add the polymerase. Molecules you will get will depend on the number of cycles, and it is directly related to 2 to the power of a traditional PCR is what? It is going to amplify your sequence, and it is going to tell you whether that sequence contains the gene which you're trying to amplify using the primer sequences that are using. So basically it's a yes or a no that you will get, and what. So you can basically sequence that sequence that you've amplified, the target sequence, and you will get to know what it is by doing BLAST and other bioinformatics tools that you can use. Now, so this is how you will analyze your polymerase chain reaction. So this is agarose gel, which I'm showing you here. This is after the reaction has been completed. These products have been run on a gel with a molecular weight marker here. And you can see that this is a ladder. Each step of this ladder is 100 base pairs, and you can see the end product. So basically it's an endpoint analysis you're doing. And this is how your product looks like when you go from 0 to 30 cycles. And you can see the intensity of the product changes over time, but it is really an insensitive way of seeing that the end product is actually changing. So here and here, if you do a densitometric scanning to see the number of products, you will see that yes, there is a difference. But if you compare between cycles here, you cannot say whether it's difference or not. So this was kind of a qualitative PCR people used to do to say yes or no. So with that, we wanted to go a step forward, and then real-time PCR reactions came into practice. Real-time by real-time, because you can see or you can monitor the reaction over time in real. So basically what is happening is in addition to knowing whether it's a yes or no and whether the reaction has that product or not, you will be able to know how much or how many starting template did you started the reaction with. So the more the starting material, the faster the reaction will occur, and that is how this real-time PCRs work. So there is a traditional way of depicting your real-time PCR reactions over time. This is how it is depicted. So this y-axis here shows you the relative fluorescence, whereas on the x-axis you have the cycle number. So if you look at this graph here, if this cartoon here, you will see that there is a baseline. This is the time during which the polymerase becomes used to the reaction conditions, and then there's a linear phase preceded by exponential phase, and after this, in this phase, your amplification of the template takes place linearly and exponentially. And after that, because the reaction conditions become limiting, you acquire something which is called as a plateau phase, right? So after this, you will no longer get any further increases in the template, the DNTPs become limiting, the primers become limiting, and the reaction stops. Now, there is a threshold line which I've shown here. This is the line which is going to cross with your exponential phase. And this is the time when your laser is going to detect a threshold amount of fluorescence. And that is called as something which is called as, traditionally called as the CT, which is your C threshold, or your CQ, which is your C quantitative value. Okay. So now when you, even if you go now, in this COVID era, everybody would have known much more about the real-time PCR. All the COVID testing has been done with the help of the real-time PCR only. And they give you the results in terms of your CT or your CQ values. So these are your CT values, the cycle values at which you can detect fluorescence above the baseline levels. Okay, so going further, how do you do your real-time PCR? What are the different ways of doing these reactions? There are two major ways in which these reactions are done. The first one is probe-based chemistry, and the second one is intercalating dye-based chemistries. Now we'll first talk about the probe-based chemistries, and the panel on the right talks about the TACMAN-based probes. So the TACMAN is a TACMAN is a copyright of the applied biosystem. So there are different other chemistries like molecular beacons which are used, but they all have a similar chemistry. So in addition to the forward and the reverse primers, we have something which is called as a probe here. So what is a probe? Probe is a sequence which is going to be complementary to your template. So when you add your primers with your probes, this probe is also going to go and hybridize to your template. Now, the difference here from the forward and the reverse primer here is that the probe contains a fluorophore as well as a quencher. In the state where it is going to be hybridized to your template, the fluorophore is going to be masked by a quencher, as a result of which there is no fluorescence which is depicted. Now, when your forward primer gets amplified by the action of the Taq DNA polymerase, the polymerization reaction takes place. During the polymerization, due to the 5' to 3' endonuclease activity of the Taq DNA polymerase, it is going to cleave this quencher, cleave this probe, as a result of which the fluorophore and the quencher get separated. Now fluorophore fluorescence is no longer masked, and you can detect this fluorescence, right? So it is the nuclease activity of the polymerase which is going to degrade this probe, and it is going to free the fluorophore from the quencher, and it is this fluorescence that you're going to detect. So more the product produced, the more the degradation of this probe, and more the fluorescence. So basically you are simply going to detect the fluorescence on the graph, right? So this thing that I'm showing you here, this graph that I'm showing you here, you will detect the fluorescence, and then you can say that yes, this is my CT value. But trust me, this chemistry is very, very expensive. Each TACMAN probe costs you around 20 to 30,000 rupees, or like $200, and it is going to be limited to a very small reaction number, say for example 200 to 300 reaction numbers, and it ends up being very expensive. So that is why people came up with a second type of reaction setup, where where they use intercalating dye. It can be cybergreen, it can be eva green. There are different chemistries which are available. This is a dye which goes and intercalates to your double-stranded DNA, right? So if you look at this part here, it is going to go and intercalate to your double-stranded DNA. And once the double-stranded DNA gets unintegrated, it starts to emit fluorescence. It also goes and binds to your single-stranded DNA, but the fluorescence which is emitted is not that strong, right? And this is a reversible reaction. So it will go and bind to your template, and it will come down to your template, right? And it binds to your minor groove. The fluorescence is much stronger when it is bound, and this is the fluorescence which is to your bound DNA that you detect on a laser of the real-time machine. So basically what is happening is, as your reaction is proceeding, your PCR product is increasing in concentration because of the number of copies being formed, as a result of which the fluorescence intensity signals go higher, and you will get the CT values. Now, the problem here is, I told you this is going to be specific. It is going to be commercially used. So here you have to design your own primers. And apart from designing your own primers, there's a lot of standardization involved. These come pre-standardized. These for the cybergreen based assays, you have to do a lot of standardizations. So how do you do a standardization? Just like you do standard curves for proteins, like when you do a BSA estimation or you do a Lowry's estimation, you have to do something similar to that here. And you take a DNA of known concentration and you do a serial dilution of that. So when I say a serial dilution, I mean that there is a 1-fold, 10-fold difference in each of these dilutions. So from the dilution, I will make a 10 to the power of 1, 2, 3, 4, and so on and so forth. I will keep diluting my DNA. And I will set up the real-time PCRs using CyberGreen with these serially diluted DNAs. Now, real-time PCR reactions are never put as a single reaction. You always have to put multiple reactions or multiple replicates for the same reaction. And people generally put it at least in duplicate, triplicate, or quadruplets. And that is why you can see multiple lines here when I talk about this. So basically what you will do is when you do these real-time PCRs with these serially diluted DNAs, you are going to get this kind of curves, right? So these are your curves. I told you this is your baseline and this is the threshold values that you're going to get. So you can see there's a CT value, or you can start detecting the amplification at cycle number 14, 17, 21, 24, and so on and so forth for different concentrations of the DNA. As the concentration of the DNA is the highest here for the orange color, which is 20 nanogram, you see it much towards the zero axis of the axis. And as the concentration decreases, the number of cycle increases as for the cycle threshold to be detected. And then you have another control here, which is referred to as your NTC, or your no template control. When I say the no template control, I mean that everything else is added into it, but your DNA. Now you're going to plot these cycle numbers onto your standard curve here, where you're going to plot quantity of the DNA that you've used on one axis, the x-axis, and the y-axis contains your CQ values. Now you will get a distribution like, you should get a distribution like this. And if you get a distribution like this is your perfect conditions. You can do your reactions in the linear range, the exponential range. So you should get, so if you see the curve declining like this, or if you see the curve declining like that, you don't work in that concentrations. I would basically choose 0.2 to 20 picograms here, because this is your linear range here, right? So and the efficiency of the reaction is very good. The R-square values are very good. So I will go with this. Now, after that, you have to do a second thing. So if you remember from my slide here, I showed you this gel, right? This is when you run the gel to analyze a traditional PCR. You can do something like this in a real-time PCR. And this is your melt curve analysis, which is done. So I told you CyberGreen goes and intercalates with your DNA, and it intercalates much more, and the fluorescence is much higher for your double-stranded DNA as compared to your single-stranded DNA. So what you can do is you can do a melt curve analysis. And when you do a melt curve analysis, you increase the temperature after the reaction is completed, after the number of cycles are done. The double-stranded DNA that you have, you will keep increasing the temperature by 1 degree, and you will see how CyberGreen fluorescence will go and decline. I'm sorry. So you will get a curve like this, which is referred to as the melt curve. This one peak corresponds to one single product that you obtain. And you will generally for real-time PCRs, the reaction size, the product size is kept between 100 to 200 nucleotides, which we'll talk about in the next few slides. And you will see that the melt curves for this size of product generally fall between 80 to 85 degrees Celsius. Now you can get peaks before this or after this. If you get peaks before this, those are also non-specific peaks, and they could be for primer dimers. So you will have to adjust your primer concentrations in a way that you do not get any primer dimers, and there should be a single peak, a single overlapping peak that you get. Also, the peaks that you see here, the amplification plots that you see here, for each DNA concentration, there should be overlapping patterns that you're using. And if you don't get that, that means that you have pipetting errors or other problems, and you need to further standardize these reaction conditions. So I've taken this from the Biorat site, and this is how, these are the characteristics which you require for a good real-time PCR reaction. So I'll just this here. Yeah. So basically your baselines here, your baselines for all the different concentrations of DNA that you've used should be relatively very flat. There should not be any disturbance here. Your plateau height doesn't matter. It can be different for different DNA concentrations, which is also expected because your temperate concentrations will also get limiting over time. Your curves should be smooth. Your dilution series should have expected spacings. So if it's a 10-fold dilution, there should be a cycle number difference of 3.3. Your replicates should be tightly clustered. Your curves should be all S-shaped, and you should not have wavy wave lines. The melt curve should have one peak per product, and if you get multiple peaks like this, you should try to standardize this even further. Going ahead with this, we'll now talk about how are these primers and probes designed. So when you do a real-time PCR, the starting material can be DNA, it can be RNA. If you're doing it for diagnosis, and if, say for example, when you talk about the COVID-19 diagnosis based on real-time PCRs, it is the RNA samples that you work with, right? So your RNA has to be first converted into cDNA, and then from the cDNA, you do the amplifications, right? So how do you design these primers and probes so that you distinguish between the RNA from the DNA contaminating your samples? You can do a DNase digestion also, but most of the times people try to save money on that. And many times your DNases are contaminated with RNases as well. So to avoid that problem, there is this very smart trick that people have evolved over time to distinguish your DNA from your RNA. And the primers and the probes that you design are going to specifically only amplify your RNA, or I mean to say that the signals that you're going to get you've designed are only going to be from your RNA counterpart and not from your DNA. So what are the different characteristics of the primers and the probes that you need to keep in mind? It should be specific to the target. So basically you can do an NCBI based blast to see if it is binding to multiple targets. If it is binding to multiple targets, it's better to design a newer primer. The primers are generally 18 to 24 base pairs in length, but depending on the type of applications, the size of the primer can be increased. Sometimes for cloning purposes, your primer can be go up to 30 to 35 nucleotides also in length. For microRNAs, if you're using stem loop primers, then it is going to be more than 35 base pairs for sure. Your product length should be 50 to 150 base pairs in length, not below 50, because then the primer dimers are going to come in that area, and it is going to give you non-specific patterns there. And not more than 200, because if it is more than 200, then cybergreen chemistry becomes a problem. It is going to be a problem analyzing that data also. The GC content should be 50% for the product. If it is more than 50%, then PCRs can be problematic to standardize, and it might be difficult to work with those primer sets. And we have to avoid GC stretches and repetitive sequence in your primers, because it becomes difficult to synthesize those primers. And if you have repetitive sequences, then the PCRs become problematic. I'll not go into the details of that, because I'll spend another 15 minutes talking about that. But just for now, keep that in mind, 50% GC content, and we have to avoid GC stretches. We have to check the primers for the dimers, especially for cybergreen based assays, because I told you that these are primers which are going to be forming primer dimers, which can be directed to your melt curves, and we do not want any primer dimers to be formed. And ideally, you should keep a 5-degree temperature difference between the annealing temperatures of the forward and the reverse primers. Now, how do you design these primers? We'll come to that trick now. So say for example, I'm working with this DNA template, and the mRNA that I'm forming is this, right? Which is depicted in this panel below. So let's first talk about primers that span an intron. These are intron spanning primers. So let's design a primer at this junction, exon junction, and at this exon junction. So P1 is going to span this exon junction, and P2 is going to span this exon junction. So the trick here is, if it is going to amplify from the mRNA or from the converted cDNA, it is going to form a shorter product here. If at all it is going to try to amplify from a DNA, no product is going to form, because the sequence which the primer is going to recognize is not present in your DNA. There's an intron which comes in between the primer sequence, as a result of which your primers won't anneal properly to your DNA, and they will not amplify. Now, sometimes it's difficult to design primers in this sequence. So that is why the second trick which is used is you design primers flanking an intron. So these are intron flanking primers. So basically you have multiple exons for this gene. I can design a primer here, and I can design a primer here. So if I get amplification from the RNA, it's going to be a 100 to 150 base pair product which I'm forming. But when the same primers try to amplify from your DNA, it is going to be a product which is going to be the size of this product 100 base pairs, say for example, plus the size of this intron. So if the size of the intron is 1 kb, it's going to be a 1.1 kb product from the DNA versus 100 base pair products from the RNA. So you can distinguish it on the basis of size, and your real-time PCR won't detect it only. The melt curves are not going to detect that kind of amplification at all. Okay, going further. Sorry. So I'm not advertising for any company here, but these are different types of reaction mixes which are available from the market. You can buy it from different companies. Most of these are going to be governed by the primer probe choice or by the choice of the platform that you're using. When I say the platform, it is a platform which is sold by different companies. Each company has its own choice, and they try that the customers buy the reaction mixes from their own company. So they have their own specifications along with it. So generally these primer mix are also available. But when I talk about the cybergreen based mixes, these are all present in 2x concentrations. So half of the reaction you can add this reaction mix, which is a consistent mix that is going to be prepared, which has pre-optimized magnesium chloride, and it works with this hot start DNA polymerase. So when I talk about a hot start DNA polymerases are having their active sites marked by a subunit which is going to be heat sensitive. So when you heat the reaction for at 95 degrees Celsius for say 10 minutes, that subunit gets inactivated and your reactions can start. So basically you use a hot start DNA polymerase to avoid any non-specific amplifications in the PCR in the process of setting up the reaction. The reaction will only get started when you have a 95 degrees for 10 minutes denaturation. So here you can use these master mixes, and you just have to add your template and your primers from outside. The remaining mix volume can be made up with nucleus-free waters. So you can do a one-step versus a two-step PCR. Most of the time people use a two-step RT-PCR because your RNA becomes limiting. And if it is a very precious sample that you're working with, say for example, a brain sample or small tissue from somewhere, it is difficult to get a large amount of RNA from these samples. So that is why people generally prefer a two-step RT-PCR over a one-step RT-PCR. So how is a one-step RT-PCR different from a two-step RT-PCR? We'll just talk about that. So here what you do is you put everything in one tube. So when I say that you do the reverse transcription, that means you convert the RNA into the cDNA in the same tube. Then you use primers which are specific to the template that you're trying to amplify over in that same reaction mix, and then you amplify that cDNA. So basically, the reverse transcriptase has a different working temperature from your Taq DNA polymerase, and you can use high temperature, which is the requirement for your Taq DNA polymerase to work, to inactivate the reverse transcriptase and activate your DNA polymerase. Now, here your RNA concentrations can become limiting. So that is why people have evolved the two-step RT-PCR. Here, you do the reverse transcription in one tube. After you do the reverse transcription, you can take the cDNA from this tube and then you set up a real-time PCR in a different tube altogether. So basically your reverse transcription would be carried out using oligo-DT or random primers. Oligo-DT, why oligo-DT? Because you're trying to amplify the mRNA from the RNA samples that you have, or you can use random primers. These are random hexamers which are going to go and bind to your RNA sequences and are going to amplify all the RNA which is present. You will require your RNA template, reverse transcriptase, oligo-DT or random primers, and the NTPs for this reaction. After this reaction is done, you can take the cDNA and you can do a PCR again using DNA polymerase, PCR specific primers, DNTPs, and your cDNA template. This is an example which I'm showing you here. This is from the Qiagen website. Again, I'm not endorsing the Qiagen website for this. It's just an example that I'm giving you. So this is your RNA, mRNA, which has this poly A tail. You can use the oligo-DT primer or you can use the random hexamers. You can do a reverse transcription. After the reverse transcription, you will get a single-stranded cDNA. You can use the RNase H function of the reverse transcriptase, which is going to degrade your RNA from this cDNA, from this duplex, which is a hybrid duplex. That is why the name H, RNase H. So you will get the single-stranded cDNA. And then from here, you can use this DNA. You can for your further reaction. So basically how this reaction proceeds is what is given here. You can mix your RNA, your DNAs, and you can add RNase-free water. You can add the reverse transcriptase mix, which contains your buffer, your RT primers, and then you incubate it at 42 degrees for 15 minutes, followed or to 2 hours, depending on what type of this reactions mix you are using, and then you can convert it at to cDNA. So this 95 degrees for 3 minutes or 85 degrees for 5 minutes is a step which is used to inactivate your reverse transcriptase. Now, there are certain controls that are used for doing these reactions. The first one is your NTC, or in your negative control, that contains your no DNA. That means everything else is there in the reaction except for your DNA. And this is used to check whether your reagents are contaminated or not. If you get abandoned in your DNA, you cannot trust that data and you will have to redo the reaction at all from the scratch. The second one is your no RT control. This is going to tell if there is any signal which is coming from a contaminating DNA. You also need to have positive controls. Positive controls are the samples which you know has the gene, and this checks whether the reagents and the primers are working. And this is very important, like in the COVID scenario. If you want to say it's a positive versus a negative, you need to have a positive control to show that yes, my reaction is working, to show the absence of the gene. Okay, so we'll now just see this video which I'm going to show you here. Wait a minute. So this is the RT-PCR tutorial, and my student Shreya, you can see here, she's trying to do the reaction here. I'll just show you how it is done. So we'll start with RNA isolation. So the tube here contains trizol. Trizol is nothing else but guanidine isothiocyanate, and it is a chemotropic detergent which is used in phenol chloroform isoamyl alcohol based nucleic acid extractions, which is going to inactivate all your DNases and RNases present in the sample. So this tube actually contained your animal cells, which she has pelleted down. And now when she is trying to mix the pellet with the trizol mix she's used, you can see the pink colored trizol is used here. And this step is going to lyse all the cells so that all nucleic acids come into the solution along with the other things. After that, you have to add chloroform. So there is a fixed protocol for 10 to the power of 6 to 10 to the power of minus to the power of 8 cells. You add 1 ml of trizol, and for every 1 ml of trizol, you will add 200 microliters of chloroform. So you can see the bottom of this tube contains this white colored chloroform. You mix it properly. This is done to separate the aqueous phase from the organic phase, and then you incubate it for 10 minutes on ice. You have to do everything in cold here because you're working with RNA samples, which are very sensitive. After this, we do a high-speed spin at 13,000 RPM for 10 to 15 minutes. And after that, you can get the nucleic acids, the RNA, in your aqueous phase. Now, the nucleic acids, I say that, so that means I mean that the DNA can also be there, but most of the times we try to do it in a way that it does not come. So you can see 2 layers being formed. This is your organic layer. We are going to work with this top layer, which is your aqueous layer. All contaminating proteins are going to come and at this interface, right? The interface between the organic and the aqueous layer. So we'll just transfer this aqueous layer slowly and carefully into another tube. You do not want any trizol to come into the tube, because if it comes, it is going to hinder the reactions forward. Your Taq DNA polymerase is very sensitive to phenol and it won't work. So we'll put it into a new tube, and now we'll try to add precipitate the nucleic acids out. So how do you do that? You generally add 0.5 volumes of isopropanol to it. One for every 1 ml of triazole, you will add 500 microliters of isopropanol, and this isopropanol will help in salting out of the DNA or the nucleic acids or the RNA in the solution, right? So this is, we'll just add this to it, we'll mix it now, and you have to incubate it at minus 20 degrees Celsius for at least half an hour. You can keep this also for overnight, because this is a step at which the nucleic acids get precipitated. After that, we do a high-speed spin to separate your nucleic acids. They form a pellet, and we'll wash this pellet with 70% ethanol, because we do not want any contaminating salts to hinder our reactions further. So why 70% alcohol? Because 70% alcohol contains 30% water, which is going to remove, or which is going to solubilize all your salts which are there. And you do not want to exceed the alcohol concentrations, or you don't want to exceed the water concentrations also, because if you exceed the water concentration, the water content, the DNA also will get dissolved in that. She's giving a wash here in this step with 70% alcohol, and it should be prepared fresh, and it should be done at you see everything she's doing in cold. So after this, you will give it a spin again in the centrifuge, and you will throw out the supernatant. The pellet, you will see a very fine pellet in this. You dry the pellet out, and then you measure the concentration of the RNA or the nucleic acid that you have isolated. So this is a nanodrop plate which we use in our lab for doing the nucleic acid estimations. So she has a sample of RNA here, which is dissolved in TE buffer, which can be dissolved in TE buffer, which is tris-10 EDTA, 1 millimolar in concentration. If you have to do long-term storage, why TE? Because EDTA in this buffer prevents the RNases from acting, and it deactivates them. But because we are going to do an amplification straight away, we are going to dissolve it in water, because TE is going to inhibit your, because it contains EDTA, it is going to chelate out the magnesium ions, and it is going to interfere with your PCR reactions also. So you do not want EDTA in the PCR reaction. So if it is going to be used subsequently for PCRs, you will immediately dissolve it in water. Otherwise, for long-term storage, you're going to use TE buffer. So she's using an underdrop here. Again, no endorsement for the product. We just want the concentrations to be known. You can use a simple spectrophotometer, which is shown here in the background, for doing these estimations. But because we are using multiple products here, we are just going to, you will get the concentration of the RNA from this sample. And after you get the concentrations by reading it at 280 nanometers, 260 to 280 ratios are important. 280 will give you the concentration of the protein, 280 will give you the concentration of the DNA or the RNA, and your 260 to 280 ratios should be close to 2, because you do not want any proteins to contaminate your samples. After this, knowing this, we'll set up the cDNA reaction. So this is a two-step PCR that we're doing. You need the RT buffer. So she's going to make a premix which contains the RT buffer, the random primers, the reverse transcriptase, and we also use the RNase inhibitor here. Why RNase inhibitor? Because we do not want any RNases to go and kill your RNA or degrade your RNA. So she's going to make up a master mix for this, and then she's going to add equal concentrations of RNA to it. So generally, for doing a 20 microliter reaction, we use 100 microliter, between 100 to 1000 nanograms of your RNA as a starting material. And these reactions are set up in these tubes, which are 0.2 ml tubes. You can see them here. Okay, what did I do? Sorry. And then she's going to mix it properly. This is the reverse transcriptase that she's showing you here. It's a temperature sensitive enzyme, and we generally try to store it at -20. We just take it out just before using it. We'll give it a nice spin. Make sure you mix the enzyme properly, because these enzymes contain glycerol, and many times they go and sit down at the bottom of the tubes, and then your PCRs won't work. After this, we'll set this up in a thermal cycler. So the thermal cycler has a particular condition. So we generally use 25 degrees Celsius for 10 minutes to activate the enzyme, followed by 37 degrees Celsius for 2 hours for converting your RNA into cDNA, and then 85 degrees Celsius for 10 minutes to inactivate your RNAs. And then, your reverse transcriptase. So this is a heated lead that we're using. If you don't have a heated lead, you can use mineral oil on the top of these tubes, and we use a heated lead to prevent any kind of evaporation which happens in these tubes. Okay, so after this, we have the cDNA samples ready. This is a Biorad thermal cycler. Again, no endorsement for the product. And this is the cycling conditions that we are using. After this, we are going to set up a real-time PCR. So these real-time PCRs are going to be set up on plates or in tubes, which are going to be having optically active surfaces. Why optically active surfaces? Because you're going to use a laser to detect the fluorescence. So that is why you should always wear gloves while handling these plates and tubes, and you should not touch anything with your bare hands. Why? Because if your hands leave impressions, the optical lasers might interfere with it, and it might create non-specific backgrounds. So this is how she's sealing it. Keep this in mind. She's not touching the seal at all. She's using a sealer here to seal this plate after setting up the reaction. The reactions, as I told you, is set up in duplicates or triplicates, and then you have to seal this properly so that no evaporation takes place. This is the Roche thermal cycler, which I'm showing you here, where we're setting up the real-time reaction. So you'll just put this up. This is a same PCR machine, but it has a laser which can detect the fluorescence. And you'll set up the PCR reaction along with the melt curve and analysis you can do. So again, she is going to now show you how the reaction looks like. So I'll just show you how this looks like. So this is your software, which is going to show you how the reaction has proceeded. So we put up a two-step amplification, 95 degrees for 10 seconds, 65 for 60, and then 97 degrees for 1 minute. This is how my reaction plate looked like. These are all my amplification curves. You can see that most of them are overlapping, and then we'll see the melt curves. So these melt curves are also overlapping. We're getting a single peak. And this is the RNA that she started with. This is how your RNA looks on the agarose gel. So these are the two bands you get because it's a eukaryotic animal cells which we have used. This is the 28S band, this is the 18S band, and this is the 5.8S band. And this small smear that you see along the wells, along the sample, that is your mRNA that you're trying to amplify by converting it into cDNA. Okay, now, so when you set up any kind of reaction, the first step that you do is you set up a standard curve. And for standard curve, you do something which is called as absolute quantification. So I told you that you do a serial dilution. After you do a serial dilution, you will do a standard curve. And then you can analyze your unknowns from plotting back onto the standard curve. So this is something which is called as your absolute concentrations. And this is what you get as real-time PCR results for your COVID PCRs. The second thing which is done for gene expression analysis is your relative quantitation. And this is a little complicated. I'll just take you through it slowly, and I hope you understand. So here we will have an untreated sample. So let's talk about comparing the effect of H2O2, which is the reagent which induces oxidative stress in cells. In untreated, so in one cells, I will not treat them with H2O2, and the other cells I'll treat them with H2O2. So my untreated will serve as my calibrator. This is my T0, and my treated one will be the one which is my H2O2 treated cells. So now you require two genes here. One would be your gene of interest. So say, for example, I'm trying to look at the expression of GPX1, which is one of the genes involved in oxidative stress. I will also need a reference gene, which was initially called as the housekeeper gene, which is abbreviated as HK gene. And this is the gene which will not show any differences in expression due to H2O2 addition. So you can calculate the CT values, the delta CT values, by subtracting the gene of interest CT values minus the housekeeping genes, and then you calculate the delta CT values by subtracting the delta CT for your sample, which is my H2O2 treated sample, versus the delta CT of my calibrator, which is my untreated sample, and then you calculate the fold change, which is 2 to the power of minus delta CT. So this is how your CT values would look like. This is how your standard curves would look like, and you will assume this is done with the assumption that the reaction is being carried out with 100% efficiency for both gene of interest and housekeeping gene. You can also substitute 2 to the power 2 with the number which is giving you the 100% efficiency, and you can do this analysis. So your real-time PCR can be used for multiple applications. You can do genes expression analysis. You can do it for disease diagnosis and management, like we have done it for the SARS-CoV-2. You can do it for food testing, how much of the food contains genetically modified parts. You can, if at all it is genetically modified, you can see that. You can do it for copy number identifications and even for forensics. So now we'll come to how it is being used for SARS-CoV-2 detection. So you would all have seen this in different forms. So this is actually a SARS-CoV-2, and it's a RNA-based virus. So the genome of the SARS-CoV-2 is shown here. So basically what people have done is they have designed these kits which are going to amplify different genes of SARS-CoV-2. You can use the spike protein gene, you can use the envelope, you can use the RNA-dependent RNA polymerase or the protease gene. And then you do the amplification for these genes. So this is basically how the reaction is done. You take a nasal swab from the site, and then you dip that swab in the virus transfer medium, via virus transfer medium, because you do not want to inactivate the virus till the RNA extraction step. After that, you inactivate the virus. You extract the RNA. Just focus on this. Don't focus on this. This is a different protocol altogether. We'll extract the RNA like I showed you, and then you will convert the RNA into cDNA, and then you will see whether the amplification of the genes for the SARS-CoV-2 is taking place. These are the samples which are tested for SARS-CoV-2, and these are my negative controls, which did not, these are my negative samples which did not have SARS-CoV-2 infections, versus these are my positive samples which were infected with SARS-CoV-2. So you can see their amplification curves also, and you will see that they were positive. Different CT values are there. Okay. So the other application of real-time PCR, you can use it for SNP detection. This becomes important when you're doing disease diagnosis for certain genes, using certain genes. So say, for example, this red G is the allele which is disease associated. So these are two people who are carriers for this allele, and you want to see whether one of the children has the disease allele or not, or he'll be infested or not. So basically what you do is you design, again, these are TACMAN based real-time PCRs, which I'm showing you here. So you use two different color probes, each specific for one of the alleles of the SNP, which is used for this typing, and you will get different colors. So basically, if you look at this plot here, these are the samples which are FAM labeled. These are the samples which are BIC labeled, and these are the samples which have both the alleles, right? So you can see this, these samples would be heterozygous for that allele, these are homozygous for the disease alleles, and these are all normal people. These are your negative controls. So this way you can use real-time PCR for allelic discrimination, which is used for disease diagnosis also. You can also use it for methylation specific PCRs. When I talk about methylation, I mean that whether a gene is being expressed or not, it will depend on the methylation status of that gene. And methylated genes are not expressed. So you can basically use a restriction enzyme, which is going to go and chew your unmethylated DNA. So your methylated DNA gets saved from being chewed. And when you do a PCR with this methylated DNA, you will see early amplification for this DNA. So you can see whether your DNAs are methylated or not. And again, this is done to check whether your oncogenes are activated, as in the case of cancers or not, or whether your tumor suppressor genes are being activated or inactivated. You can also use it for telomere length measurements. This is an assay which is done again to see whether your cells have acquired a cancerous phenotype or not. So basically your cancer cells have longer telomeres as compared to your normal somatic cells, which see shortening of telomeres as the cells divide. So you can convert, again, if it is a longer telomere, you will get higher signals using qPCR versus a shorter telomere, again based on the fluorescence. And you always have to normalize with a gene which has a single copy number, and based on the values you can actually see. And I'll stop here by acknowledging my lab here. So we were trying to take a picture here yesterday, and I like this picture more because we are all happy. It's a happy lab environment that we have in our lab. Devika and Shraddha helped me to make this video, which I've shown you here. Shantanu and Pradnya are MSc dissertation students. Pratiksha is another PhD student in the lab. Gurvinder and Praveen are MSc students from the department, and we have a very rich lab alumni. I'm showing you a few names here. The lab funding comes from DBT, UGC, and Savitribai Phule Pune University, and these are my collaborators. And I would like to take the last minute here advertising for a book that we have published with Elsevier, which is referred to as Prognostic Epigenetics. If at all somebody's interested in studying about epigenetics and how it is used for disease management, you can refer to this book. And thank you. I'm open for any questions. Hopefully I finished on time. Hello.
Hi, ma'am. Yes. Thank you so much. It was an amazing talk. I think I also clearly understood the concepts of real-time PCR. It was really amazing session. So thank you for this very informative session. I'm sure it was a very thought-instigating one session for everyone present. We will now have a 10 minutes Q&A session. Ma'am, we can check the YouTube comments, and then if they have any questions, we can always clear them out.
Yeah.
Meanwhile, while the questions are coming in, I would like to thank all the attendees of the session too. We hope you're taking home a good lesson of learning real-time PCR from such an expert. People who missed out, please do not worry. The live session will be uploaded on our YouTube channel soon, and you can always go back and clear out your concepts. Also, to get notifications, please subscribe to our YouTube channel and push the bell icon so you get the notifications. Also, we have a Python and BioPython courses available on our YouTube channel. So subscribe our channel and start learning. And also, you can get its free certificate. Though it's a limited offer, and the channel will be and the videos would be locked in 2 months. So please, do it as soon as possible, and you will be getting free certificates for it as well. Also, I would like to update about some other projects that we're working on. So we have a 21-day NGS course that's coming up. So there'll be hands-on training. It's a 21-day online course with hands-on training. The classes are starting from, the first batch is starting, in fact, tomorrow. And apart from this, we also have 2 workshops coming up on proteomics pipeline and qACR modeling. So check out our website, you'll get all the details there. Lastly, LifeGenBio believes in empowering your careers and also offers personalized services and trainings in bioinformatics and life science subjects. We have 4 such ongoing projects. If you're interested, do let us know. We'll be happy to provide any services that we can. Lastly, just wanted to tell you guys to share your valuable feedback. I've shared the feedback link in the comment section. So do share your feedback with us. And yeah, I think that's it. So thank you. You can always reach out to us. And I think, yeah, that's it. Tanev, you can take over.
Okay, so there is one question here. How can we fix the primer to particular virus, and how many cycles require for COVID-19? So can I start the answers? Hello?
Yes, ma'am. Yes. You can go on with
So,
that.
so to answer this question, so you need to design primers which are specific for your COVID-19 genome. So the genome sequence is already known. It has been deposited in different databases. You will design the primers specific to that sequence, and you will take areas of the sequence which are not going to be specific to any other virus. So it's going to be specific to only COVID-19 strains. And for COVID-19, generally we try to take up to cycle number 40. But if the CT values are detected above cycle number 35, we generally don't take that into account, and you do a retesting. So that is why these people are asked to go for a retesting. So hopefully I answered this question. The next question is it necessary to do DNA treatment before cDNA preparation? So as I told you before, many people avoid this DNA treatment step because the DNases might also contain the RNases, which are going to degrade your RNA. So we generally use primers, which are made either spanning an intron or flanking an intron, and either in the exon-exon boundaries or with an intron between the two exons, so that there is no problem with the contaminating DNA as well. The next question is, how can we fix, RNA viruses are either positive stranded or negative stranded. Does it make a difference in reverse transcription PCR and qPCR? No, it doesn't. No. You can design your primers and probes specific to the sequence you're trying to amplify, and it should be fine. It doesn't matter. You can use even DNA for doing quantitative PCRs. Like I showed you for the telomeres and for the SNP detection, we use DNA samples. Then how can we detect single nucleotide defect through RT-PCR if we are using a primer or probe which are 15 to 20 nucleotides? Okay, so basically when you do use a primer or a probe, when you do a SNP detection using real-time PCR, you use probes of two colors. One is going to be specific for one allele. The second is going to be specific for the other allele. And the probe is going to be made in a way that it is only going to hybridize to the A allele or the T allele. Let's talk about the one allele or the other allele. And the 3' and the 5' ends are matched in a way that it is going to be specific to that particular SNP itself. So it will not be amplified it will not be hybridized if it is not going to be that sequence, right? So we make the probes in that way. Is that fine? Did I answer that question? How can you isolate mRNA of COVID-19 from millions of bacteria and virus? Because SARS and MERS are 99% similar. So even if they are 99% similar, you can use multiple combinations of genes. So that is why when you do a COVID-19 testing, we do not do it with using a single gene. We do it with at least 3 genes at a time. So that we get information for the SARS-CoV-2 virus. And even if they are 99% similar, there might be regions of the genome which are 1% different. We can design the primers from that region, and we can be sure that it's the SARS-CoV-2 virus that you're amplifying, not the MERS. Okay. Any more questions?
No, ma'am, I think that is all for today.
Okay.
More questions in the chat box.
Okay.
Yeah, so I would like to thank you, ma'am. It was a great session. We all loved it.
Okay.
And Ankita, yes, now you can circulate any more information you want to.
No, I think that's it. Just wanted to tell them that the feedback form is in the comment section. Tane, if you can pin it, so that will help the guys. And I think that's it, ma'am. You were great. The session was amazing.
Thank you. Thank you for having me. And if you want me again, I will be happy to give more talks.
Definitely, ma'am. Definitely.
Yeah. Okay. So should I stop the sharing?
Yeah, sure.
Okay.
Thank you, everyone.